In this video from 2 years ago, entitled Darwinian Materialism versus Quantum Biology, it was shown that Darwinian evolution, with its reductive materialistic framework, is not even on the correct theoretical foundation in order to properly understand our present findings in quantum biology in the first place. And in this following video that I made from 2 months ago, entitled How Quantum Mechanics and Consciousness Correlate, it was shown that a tremendous amount of information must be coming into a developing embryo from the outside the embryo by some non-material method. In this present video, I would like to improve on the first video that I made 2 years ago and to incorporate the information from the video that I made 2 months ago in order to hopefully improve the video and make the argument that I made in the first video much stronger than it originally was. In the first video, it was shown that over the last several decades, Darwinists, because of their reductive materialistic framework, have presupposed there to be far more random thermodynamic jostling of atoms and molecules in molecular biology than there actually is turning out to be. For instance, in the following 1998 article, Bruce Alberts, who was a 2-time president of the National Academy of Sciences, stated that we have always underestimated cells, but at least we are no longer as naive as we were when I was a graduate student in the 1960s. Instead of a cell dominated by randomly colliding individual protein molecules, we now know that nearly every major process in a cell is carried out by assemblies of 10 or more protein molecules. And even though Dr. Alberts wrote that article 21 years ago in 1998, Darwinists, because of the presuppositions they hold within their reductive materialistic framework, are still very much reluctant to let go of their belief that the cell is dominated by randomly colliding individual protein molecules. For a prime example of this fact, in 2006, Harvard University, via a production company called BioVisions, made a video which was entitled The Inner Life of the Cell. The video by Harvard BioVisions was one of the first videos on the web that animated some of the amazing molecular machines that are now being found in cells. As you can see, the overwhelming impression of intelligent design of the cell literally leaps out of the video at you. And since the intelligent design of the cell is readily apparent for all to see, Dr. William Dembski, one of the founding pioneers of the intelligent design movement, would, around 2007, show the video in some of his lectures to students on intelligent design. When Harvard BioVisions caught wind of Dr. Dembski using the video in his lectures to his students, they warned him to not use their video anymore. Their Harvard BioVisions effort to stop William Dembski from was futile, though, since the video soon went viral on the internet, and anyone with access to a computer could download the video and watch it whenever they wanted to, and see for themselves the amazing design that is readily apparent. The Darwinists at Harvard BioVisions, who had originally made the video, apparently did not like this development one bit. and in 2013, apparently trying to undo the damage that was done to Darwinian thinking by their original video, Harvard Biovisions then made a subsequent video that was entitled Inner Life of the Cell, Protein Packing. Now, in the 2013 video, as you can see, Harvard Biovisions tried to make the inner workings of the cell look as random, chaotic, and haphazard as they possibly could, so as to try to dispel any impression of intelligent design that they had inadvertently created in the first cell. In fact, in 2014, the New York Times itself ran an article on the protein packing video from Harvard Biovisions. I'm sure that many ID advocates wish they could get free promotion for their videos on intelligent design in the New York Times. But anyways, in the article, Carl Zimmer stated that in 2006 version of the video, we can't help seeing intention in the smooth movements of the molecules. But of the 2013 video, he said that the molecules of the cell flail blindly in the crowd, and went on to say, our cells work almost in spite of themselves. Thus, even though Dr. Bruce Alberts himself, all the way back in 1998, had largely dispelled the myth that the cell was dominated by randomly colliding individual protein molecules, nonetheless, Darwinists, as late as 2014, and motivated primarily by their bias against the conclusion of intelligent design, were still widely disseminating the false claim that the cell was dominated by randomly colliding individual protein molecules. Yet, regardless of their overt bias against anyone daring to see intelligent design in the cell, the fact of the matter is that we now have abundant evidence firmly establishing the fact that the cell is not nearly as random and haphazard in its makeup as the Darwinists at Biovisions would prefer people to believe. For instance, in the following article from 2014, Dr. Jonathan Wells takes direct issue with Carl Zimmer's false claim that biological molecules are flailing blindly in the crowd, and states, but that's not what the biological evidence shows. In fact, kinesin moves quickly with precise movements to get from one place to another. Moreover, in the following 2016 paper, it was found that crowding in cells doesn't hamper protein as much as they thought it did. In fact, finding a lack of collisions in the cell, in the crowded cell, was a counterintuitive surprise. Specifically, one of the researchers stated, this was a surprise. It's counterintuitive because one would think collisions between a protein and other molecules on DNA would slow it down, but the system is so dynamic it doesn't appear to be an issue. In fact, instead of biological systems being dominated by randomly colliding individual protein molecules, the fact of the matter is that biological systems are now shown to be extremely resistant to the random background noise of a cell. As the following article on photosynthesis stated, these biological systems can direct a quantum process in astoundingly subtle and controlled ways. showing remarkable resistance to the aggressive, random background noise of biology and extreme environments. Likewise, the following article on human vision stated that research has shown that humans can detect the presence of a single photon, the smallest measurable unit of light. It is remarkable a photon, the smallest physical entity with quantum properties of which light consists, is interacting with a biological system consisting of billions of cells, all in a warm and wet environment. And the researcher added, the response that the photon generates survives all the way to the level of our awareness, despite the ubiquitous background noise. Any man-made detector would need to be cooled and isolated from noise to behave the same way. What we want to know next is, how does a biological system achieve such sensitivity? How does it achieve this in the presence of noise? Moreover, in direct contradiction to Carl Zimmer's claim of molecules flailing blindly in a crowd, in the following article subtitled How Biomolecular Machines Can Generate Non-Trivial Quantum States, the author states that entanglement can be maintained even in the presence of very intense noise. And in the following articles, the author even goes on to state that this reverses the previous orthodoxy which held that quantum effects could not exist in biological systems because of the amount of noise in these systems. Environmental noise here drives a persistent and cyclic generation of new entanglement. Thus, instead of the molecular machines of the cell and the molecules being dominated by the random noise in the cell, as Carl Zimmer had possibly imagined in his New York Times article, the molecular machines of the cell are instead shown to have remarkable resistance to the aggressive random background noise of biology in extreme environments. Moreover, molecular machines are apparently designed in such an ingenious way so as to feed off the noise of the cell. Quote unquote, environmental noise here drives a persistent and cyclic generation of new entanglement. Moreover, in the following microscopic pictures of the bacterial flagellum, you can see for yourself that the molecular machines of the cell are not nearly as chaotic and haphazard looking as the Harvard's protein packing video had falsely tried to portray them to be. Even protein molecules themselves that make up the building blocks of the molecular machines of the cell do not find their final folded form by the random collisions of particles within the cell. As the following article states, when one calculates the number of possible topological configurations for the amino acids in even a small, say 100 residue unfolded protein, random search could never find the final folded conformation of that same protein during the lifetime of the physical universe. And yet, proteins obviously do not take the lifetime of the universe to randomly find their final folded form. In fact, most small proteins fold on a millisecond and even microsecond timescale. Thus, however proteins themselves may be managing to find their final folded form, they obviously are not being hampered in any significant way by the randomly colliding particles in the cell. In fact, in the following articles, the authors found that the long-standing mystery of exactly how a protein is able to find its final folded form so quickly can easily be explained if protein folding is allowed to be a quote-unquote quantum affair, where the protein could jump from one shape to another without necessarily forming the shapes in between. Moreover, the following 2015 article experimentally confirmed that proteins are indeed based on quantum principles. More specifically, the following study observed that quantum processes concentrate all the vibrational energy in a biological protein into its lowest frequency vibrational mode. Thus, proteins are apparently ingeniously designed in such a way so as to be very resistant to any random noise that may be present in the cell. As well, in the following 2006 article entitled Classical and Quantum Information Channels in Protein Chain, it was stated that on the basis of force constants, displacements of each atom in peptide plane, and time of action, we found that the value of the peptide plane action is close to the Planck constant. This indicates that peptide plane, from the energy viewpoint, possesses synergetic classical quantum properties. And in this following more recent 2015 paper entitled Quantum Criticality in a Wide Range of Important Biomolecules, it was found that most of the molecules taking part actively in biochemical processes are tuned exactly to the transition point and are critical conductors. And the researchers further added that finding even one biomolecule that is in the quantum critical state by accident is mind-bogglingly small and, to all intents and purposes, impossible, of the order of 10 to the negative 50 of possible small biomolecules, and even less for proteins. As well, DNA itself does not belong to the world of classical mechanics, but instead belongs to the world of quantum mechanics. In the following video, at the 22.20 minute mark, Dr. Riper shows why the high temperature of biological systems do not prevent DNA from having quantum entanglement. And then at the 24-minute mark, Dr. Riper goes on to remark that practically the whole DNA molecule can be viewed as quantum information with classical information embedded within it. Thus, quantum coherence and/or quantum entanglement, contrary to what was believed to be possible in molecular biology just a few short years ago, is now found to be ubiquitous within molecular biology. To repeat, quantum effects are now found to be in all the important biomolecules of life. Moreover, these quantum effects and biology are also shown to be extremely resistant to the random background noise of the cell. This is exactly the opposite type of biological activity that the Harvard protein packing video is trying to portray. On top of that, what else is so devastating to Darwinian presuppositions with the finding of pervasive quantum coherence and entanglement within molecular biology is that quantum coherence and entanglement is a non-local, beyond space and time effect that requires a beyond space and time cause in order to explain its effect. As the following paper entitled "Looking Beyond Space and Time to Cope with Quantum Theories" stated, our results give weight to the idea that quantum correlations somehow arise from outside space-time, in the sense that no story in space and time can describe them. Moreover, as the following study found, the greater the number of particles in a quantum hypergraph state, which is exactly the type of quantum coherence that we're dealing with in protein and DNA molecules, the more strongly it violates local realism, with the strength increasing exponentially with the number of particles. Darwinists, with their reductive materialistic framework, simply have no beyond space and time cause that they can appeal to so as to be able to explain the non-local quantum coherence and entanglement of the cell. Whereas, on the other hand, Christians readily do have a beyond space and time cause that they can appeal to, as has been stated for thousands of years in Colossians 1:17. He is before all things, and in him all things hold together. As to the observation of classical information being embedded within the quantum information of DNA and proteins, and since the main argument of the entire ID versus evolution debate centers on the sheer inability of random Darwinian processes to generate classical information, it is also very important to note that classical information is now shown to be a subset of quantum information. In making this point clear, it is first important to note that quantum entanglement is critical for quantum information processing to even be possible in the first place. As the following article states, quantum entanglement plays crucial roles in quantum information processing, such as quantum computation, quantum teleportation, dense coding, quantum error correction, quantum cryptographic schemes, entanglement swapping, and remote states preparation. And as the following article states, quantum entanglement is a physical resource, like energy. A pair of quantum systems in an entangled state can be used as a quantum information channel to perform computation and cryptographic tasks that are impossible for classical systems. And now, this classical information that is the central focus of many arguments between intelligent design proponents and evolutionists is shown to be a subset of this non-local quantum information by the following method. Specifically, in the following 2011 paper, researchers show that when the bits in a computer to be deleted are quantum mechanically entangled with the state of an observer, then the observer could even withdraw heat from the system while deleting the bits. Entanglement links the observer's state to that of the computer in such a way that they know more about the memory than is possible in classical physics. In measuring entropy, one should bear in mind that in quantum information theory, an object does not have a certain amount of entropy per se. Instead, an object's entropy is always dependent on the observer. Moreover, in the 2010 realization of the Maxwell-Demon thought experiment, which was originally devised by James Clerk Maxwell all the way back in 1867, it was finally experimentally realized in 2010, to repeat myself. And as the following paper highlights, it has now been experimentally demonstrated that knowledge of a particle's location and or position converts information into energy. And as the following 2010 article stated about the preceding experiment, this is a beautiful experimental demonstration that information has a thermodynamic content. And as the following more recent 2017 article stated, James Clerk Maxwell said, "The idea of dissipation of energy depends on the extent of our knowledge. Quantum information theory describes the spread of information through quantum systems. 15 years ago, we thought of entropy as a property of a thermodynamic system," he said. Now, in quantum information theory, we wouldn't say entropy is a property of a system, but a property of an observer who describes a system. Again, to repeat that last sentence. Now, in quantum information theory, we wouldn't say entropy is a property of a system, but a property of an observer who describes a system. That statement about entropy being a property of an observer who describes a system, for anyone involved in the intelligent design versus Darwinism debate, ought to send chills down their scientific spine. Simply put, these developments go to the very heart of the ID versus evolution debate and directly falsify, number one, the Darwinian claim that immaterial information is merely emergent from some material basis, and number two, these experimental realizations of the Maxwell-Demon thought experiment go even further and also directly validate a primary claim from ID proponents that an intelligent designer who imparts information into a biological system is necessary in order to circumvent the second law of thermodynamics. As William Dembski himself stated all the way back in 1999, it is complex specified information that enables Maxwell's demon to outsmart a thermodynamic system tending towards thermal equilibrium. To more clearly illustrate just how all this plays out in the ID versus evolution debate, it is first important to note that, as I made clear in the following video entitled Darwinism versus Biological Form, that Darwinists have no real clue how any particular embryo might achieve its final adult form. Within their theory, Darwinists hold that the basic adult form of any given embryo is ultimately reducible to mutations in its DNA. Yet, the failure of reductive materialistic explanations to be able to explain the basic form of any particular organism occurs at a very low level. much lower than mutations to DNA itself. The following article, entitled Quantum Physics Problem Proved Unsolvable, Golden Turning Enter Quantum Physics, which studied the derivation of macroscopic properties from a complete microscopic description. The researchers remarked that even a perfect and complete description of the microscopic properties of a material is not enough to predict its macroscopic behavior. The researchers further commented that their findings challenge the reductionist point of view, as the insurmountable difficulty lies precisely in the derivation of macroscopic properties from a microscopic description. Thus, not only do Darwinists presently have no clue how an embryo might achieve its adult form, since their theory is based on reductive materialism, it is in fact impossible for them to ever find a real clue as to how an embryo might achieve its adult form. Now, don't get me wrong, there are very many books on embryology with a plethora of very technical details as to what and when things are happening in a developing embryo. But just because we have a lot of knowledge about what and when things may be happening in a developing embryo, that still does not answer the primary question we have of exactly how do these things happen within a developing embryo. As Alexander Sierras makes clear at the 7.5-minute mark of the following video, the magic of the mechanisms inside each genetic structure saying exactly where that nerve cell should go, the complexity of these, the mathematical models on how these things are indeed done are beyond human comprehension. Even though I am a mathematician, I look at this with a marvel of how do these instruction sets not make these mistakes as they build what is us. It's a mystery, it's magic, it's divinity. As to providing us with a real clue as to exactly how an embryo might actually achieve its adult form, in the following video at about the 41-minute mark, Dr. Jonathan Wells, who happens to specialize in embryology, using a branch of mathematics called category theory, demonstrates that during embryological development, positional information must somehow be coming into the developing embryo from the outside by some non-material method in order to explain the transdifferentiation of cells into their multiple different states during embryological development. The amount of positional information that is somehow coming into a developing embryo from the outside by some non-material method is immense, vastly outstripping by many orders of magnitude the amount of sequential information that is contained on DNA itself. As Doug Axe states in the following video, there are a quadrillion neural connections in the human brain. That's vastly more neural connections in the human brain than there are bits of information in the human genome. So there's got to be something else going on that makes us what we are. And as the following article states, the information to build a human infant atom by atom would take up the equivalent of enough thumb drives to fill the Titanic, multiplied by 2,000. As to how thermodynamics itself relates to this immense amount of positional information that is somehow coming into the developing embryo from the outside by some non-material method. Work done on bacteria can give us a small glimpse into just how far out of thermodynamic equilibrium multicellular organisms actually are. The information content that is found to be in a simple one-cell bacterium, when working from the thermodynamic perspective, is found to be around 10 to the 12 bits, which is equivalent to about 100 million pages of the Encyclopedia Britannica. In comparison, the largest libraries in the world have about 10 million volumes, or about 10 to the 12 bits. Thus, since bacteria cells are about 10 times smaller than most plant and animal cells, and since there are conservatively estimated to be around 30 trillion cells within the average human body, then that gives us a rough ballpark estimate of around 300 trillion times 100 million pages of the encyclopedia, or about 300 trillion times the information content contained within the books of all the largest libraries of the world. Needless to say, that is a massive amount of positional information that is somehow coming into a developing embryo from the outside by some non-material method. On top of all that, and as was highlighted earlier, as far as quantum information theory is concerned, this positional information that is somehow coming into the developing embryo from the outside by some non-material method to bring the developing embryo to be so far out of thermodynamic equilibrium in the first place is found to be, quote unquote, a property of an observer who describes a system. In other words, some outside observer, due to the quantum non-locality of the quantum coherence and entanglement in biological molecules, must necessarily exist outside the space-time of the universe itself. And this outside observer is now required in order to give us an adequate causal account so that we may be able to explain how it is even possible for this immense amount of positional information to be coming into a developing embryo from the outside by some quote-unquote non-material method. And whereas Darwinists, because of their reductive materialistic framework, denied the existence of anything beyond space and time, especially denying God, on the other hand, Christianity just so happens to give us an adequate causal account for exactly who this outside observer might be who is imparting this immense amount of information into a developing embryo. As Hebrews chapter 4 verse 13 states, "And no creature is hidden from his sight, but all are naked and exposed to the eyes of him to whom we must give account." And as Psalm 139:13-14 states, "For you formed my inward parts, you covered me in my mother's womb. I will praise you, for I am fearfully and wonderfully made." One final note. It is also important to realize that quantum information is conserved, as the following article states. In the classical world, information can be copied and deleted at will. In the quantum world, however, the conservation of quantum information means that information cannot be created nor destroyed. The implication of finding non-local, beyond space and time, and conserved quantum information in molecular biology on such a massive scale in every important biomolecule is fairly and pleasantly obvious. This pleasant implication, of course, being the fact that we now have very strong empirical evidence suggesting to us that we do indeed have an eternal soul that is capable of living beyond the death of our material bodies. As Stuart Hameroff states in the following video, the quantum information isn't destroyed. It can't be destroyed. It's possible that this quantum information can exist outside the body, perhaps indefinitely, as a soul. Well, that's the end of the video. Thanks very much for watching. And again, all papers and videos referenced in this video may be accessed in the link that is provided in the video description. Thanks again for watching.